going to give a couple announcements. So I'm a member of the MAPS committee, so just a couple things that are coming up that we want you to know about would be Science Night is coming up on November 7th. And then our next MAPS talk is on November 14th, and that talk is going to be on CRISPR, so that should be a really interesting one as well. And it's my honor to be able to introduce our speaker for tonight. She comes from UC Davis, and it's Dr. Karen Moxon. She is professor and co-director in the Center for Neuro-engineering and Medicine at UC Davis, and she's here to share her very exciting research that she works on. So we can give her a warm welcome.
So thank you very much for the introduction, and it's a great pleasure to be here. I'm so impressed with so many people here on a Friday night. It was late, but it's really nice to see everybody. So yes, neuroengineering is a field that's been around now maybe 20 years. Mostly started with restoration of sensory motor function, and today I'm going to talk to you about how it's moving more into the field of cognitive neuroengineering. So when I first began my career, we were very interested in, can we interface with the brain? How can we put sensors into the brain so that we could record the signals and try to understand how the brain functions? So I worked a lot on projects developing little micro devices that could amplify and filter the signal. I worked on the development of microelectrodes with certain coatings so that they could be detected when they're inserted into the brain. Because the brain has a bunch of cells, but the cells are all living in a salt solution. And imagine if you put something metal in a salt solution, it's going to rust, it's going to decay. We had to protect the microelectrodes for this type of application. We wanted to make them more biocompatible. So also inside the brain, besides your immune cells, when something foreign comes in, they come and attack it, so that glial cells would all wrap around the electrodes and you couldn't record from them anymore. And we found that if we built the electrodes with nanostructural surfaces, it mimicked the extracellular space better. The immune cells didn't bother it so much, and these electrodes could go in the brain and we could record for very, very long periods of time. These were very rigid, and so then we moved on to try to create microelectrode systems that were on thin films and very flexible. And one of the big applications was to be able to stimulate in order to restore function into the body. And quickly after that, we moved to, well, what if we stimulate into the brain and then can control what animals are doing? We could begin to better understand how the brain encodes these things itself. And so now we're looking at the development of closed-loop systems. This is an example of a closed-loop system for seizure control, where you would put microelectrodes in the brain, you would record the signal, you'd process the signal, you would follow the data over time and try to predict when a seizure was going to occur, and then you could stimulate back into the brain and prevent that seizure. So these are the kinds of things that I've been working on. So just to get a little bit more granular, what are we really talking about? Well, inside the brain, you have a large population of neurons. If you stick a microelectrode in there, you'll record an electrical signal that's sort of some summation of the activity going on around the tip of your electrode. These neurons are all very close together, so you can record activity from more than one neuron at a time. And each of these little spikes here would be a single neuron sending an action potential that says it's sending information out. And if you just take, like, let's say one of these, and you glue it up in time, it would have its own unique shape. So now this would be a single neuron firing an action potential, which says that that cell is sending an output to another cell. And what we really want to know is, when did that cell fire an action potential? So we just keep track of what we call its spike times, or every time it fires one of these action potentials, we just put a tick mark. And so our complex analog signal here just becomes a series of zeros and ones. A 1 every time the cell fires an action potential, and otherwise it's 0. And I only care about the rate at which the cell is firing, okay? So what I want to talk to you about today is review sort of the history of brain-machine interfaces. So how many people know about brain-machine interfaces? So I go, not too many, just a little tiny bit, okay. So I'll talk to you about what brain-machine interfaces are, how they work, and the value that they bring, both in terms of clinical applications, but also being able to use to understand how the brain encodes information. And then I'm going to switch gears a little bit and introduce a new field of cognitive neuroengineering. So it's very similar to this brain-machine interfaces, but we're using it to understand higher cognitive processes. And in the example I'm going to use today, we're using it to understand how people make decisions. And I'll show how it can be used to understand how the brain encodes information, and then some clinical applications that we could use this for. So, brain-machine interfaces, I happened to be extremely lucky when I was a postdoc. I got to work with some brilliant people who were trying to develop the first brain-machine interface. And basically how it works is, we trained an animal to press a lever with his little paw there, and as the animal pressed the lever down, a little robot arm would go out, grab a drop of water, and as he released the lever, the arm would come back and he could drink his water. So we trained the animals very well, we'd make them a little bit thirsty, and they would continually go into the little chamber and they would press the lever, the arm would go out, get a drop of water, they'd release the lever, and the animal would be able to drink his water. So they would be doing this over and over and over again, and we would implant the animal with arrays of microelectrodes into the brain and record the neural activity from across all those different channels. So each of these is an example of the cell firing and action potential. And then, as I said before, for each cell, we would look at the time at which that cell fired an action potential. And we know that neurons fire in response to movement if we put them into the motor cortex, but no one cell does a very good job representing the animal pressing his paw down and back up. But if we combine the activity over hundreds of neurons, we can get a fairly good representation of the animal pressing the lever down and then releasing it. So we're recording from lots of cells, we're combining the activity across all these cells and coming up with a population function that describes how the animal's moving. So as the animal's doing this task, about halfway through it, we make a switch. And now no longer does this lever arm control the robot arm. Now we have the neural signal control the robot arm. So as the animal is thinking about moving his arm, that goes up and moves the robot arm out and then back. And so those are the first signs of brain-machine interfaces. Can we use your brain, take the information, decode it, and have it do useful work in the outside world? So just to give you some little insight into kind of how this works and how we get this population function, as I noted before, different cells fire action potentials at different times, and we're keeping track of when those cells fire. We have maybe 100 microelectrodes implanted, so not just 3 cells, but let's say 100 cells. And you could imagine now taking a multidimensional space, in this case it's 3-dimensional space, and for every moment in time saying, well, what is the firing rate of the cell in this neuron space? And so you would get a data point that would just give you the firing rate of each of the cells and give one data point. Then you would move this along in time, and for every moment in time, you get a new estimate of how the firing rate of the cells are changing in this sort of multi-neuron space. And we sort of call this movement through here a neural trajectory. So it's how the neural activity is moving in this multi-dimensional neural space. But because we have so many neurons, I can show you this in 3-dimensional space with just 3 neurons, but you couldn't imagine it if you had 100, right? And we want to really understand. So we need to somehow take this high-dimensional space and condense it down to a lower-dimensional space that still carries the information we're looking for. So what we're trying to do is basically find sort of a new set of axes that can still describe all of this activity, and the new space is defined by these what we call latent variables, and they come from this model that we develop of the neural system. So just conceptually, if you can kind of understand, I can take this neural activity, I can use it to develop a model that only has a few variables in it, and then I can plot this neural activity all in this lower dimensional space, but not lose anything because it's allowing that data to be collapsed to a smaller dimensional subregion. So that's kind of comfortable and kind of a overview type of way. I'll just try to go through a little more detail and try to fill in some of the blanks there. So we know we have this lever movement. So we know that the animal pushes the lever down and then he releases the lever. He pushes the lever down and he releases the lever. And what we want is this population function that's going to mimic this movement. So as I noted before, for any single neuron, there really isn't a very good correlation between the individual neuron firing its action potential and the activity of the paw. Not too bad, there's some information there, but each individual neuron by itself doesn't give you very much information. However, as I mentioned, if we combine the activity across all of these neurons, we can create a model that has a latent variable that does a fairly decent job. So the model in this case is something called principal components analysis. And what principal components analysis is, it just asks, how does the activity across all these neurons co-vary? And I'll take the most amount of variance in this neural activity. And so it's just looking at how the neurons all fire together. But we know that activity in the brain is not just linear, so principal components analysis is a linear model. We know that it's nonlinear, so then we went to little artificial neural networks, or a recurrent neural network here, and used this neural activity to train the artificial neural network, and it could do a very good job reproducing the lever movement in time, so we could almost perfectly reproduce the animal's desire to press the lever whenever he wanted to and get his reward. So these nonlinear latent variables, so this would be a latent variable, this would be a latent variable, outperform linear variables. So this is about an 86% correlation, this only has a 50% correlation. So this is one way in which we can use brain-machine interfaces to try to understand how the brain encodes this information. So the advantage of this approach is it goes beyond traditional correlative analyses that you see in neuroscience, where people are just asking, oh, if the neurons go up, if the animal moves, does the firing rate of the neurons change? This is actually allowing us to be able to reconstruct what the animal is doing with his behavior. It enables a quantitative metric of the amount of information that's represented by those neurons. So the better job I do reproducing how the animal moves, then that's more information I have about how the brain is encoding it. So it can be used to reveal how the neurons work together to convey that information, and in some ways kind of explain the computations that the brain is doing to perform motor control. So this very quickly was looked at as a new way in which we could study how the brain works. And very quickly people started doing it in non-human primates. So having them control the robot arm and take up a piece of banana here, get some banana. And then very quickly went to humans. And there was a project that started in the early 2000s, a project called BrainGate. And basically people who were paralyzed from the waist down could be implanted with these microelectrodes into their brain and use it to control a cursor on the computer screen. And this is an example of a person controlling a robot arm, and they're moving the robot arm in real time to contact each one of these targets. So there's still an enormous amount of work going on in this field. There's a whole new neurotechnology industry. This is just some of them doing some work in this area. If you want to get a good fuller list, you can go to Dr. Singer's website at Georgia Tech, and he keeps a very nice up-to-date website in all of these new companies working in this space. So at this point, I began to be interested not just in motor movement, but I was very interested in how the brain makes more complex activities. And especially I was interested in decision making. So how is it that we make the decisions we make, and how does the brain process information in order for us to make those decisions? So the example that I was very interested in was risky decision making. And one of the reasons for that is because on either end of the spectrum you have some pathologies, right? If I am too enamored with making risky decisions, then I'm more likely to gamble or do things that might not be in my best interest. If I'm too risk averse, then it can actually create an environment where I'm not willing to do things that regular people are willing to do. Right? I don't want to go outside, I don't want to talk to people, I don't want to interact, and it can be part of a sort of more example of someone being on the depression scale. So what we did was we came up with a game that people could play where they could either take a safe bet or a gamble bet, and while they were doing that, we recorded neural activity from their brain. So we're specifically interested in making decisions under conditions of risk. And so for an example of how this is going to work is, are you the kind of person who wants to eliminate risk and only take a sure thing, something like the bird in the hand? Or are you someone who would like to risk the sure thing to go for something better? And that's kind of along the lines of fortune favors the bold, so people who are willing to risk what they have to go try something new, fortune can favor you. So this is the kind of task that we're going to ask people to do, and then we're going to record the neural activity from their brain and see how the neural activity changes as they make these different types of risky decisions. What we want to know is, can we predict someone's choice before they actually indicate their decision? And if so, what are the neurophysiological mechanisms that underlie how they make these decisions? And then can we change someone's propensity to make risky decisions using some kind of neural stimulation or neuromodulation? So I teamed up with my colleague Ignacio Sayas, who's a professor at Mount Sinai in New York, and he had this very interesting task. So the way the task works is you're given a number from 0 to 10, and you are asked to assess the likelihood that the next number will be smaller. So, they're pulled from a uniform distribution, so you're equally likely to get any number from 0 to 10. And your options are two. First, you can take a safe option. It's fixed $10 regardless of the outcome of the trial. Alternatively, you can gamble. So if you think the second number is going to be less than the first, you could get up to $30. But if the second number is less than the first, should be greater than the first, then you'll get nothing. So you either take a safe bet, you get $10 no matter what, or you take a gamble, and if the next number is bigger, you'll get the $10 or the $30, otherwise you'll get nothing. So the gamble is all or nothing, or the safe option. So people do this task on a regular basis. This is what we refer to as someone's psychometric curve. So if the number is very small, then you're not very much likely to take a gamble bet because the next number is very likely to be higher, so you'll always take a safe bet. If the number is very large, the next number is very likely to be small, so you're much more likely to gamble. And then, of course, as you get around the middle, the change in whether or not you gamble or don't gamble is it changes rapidly. So if the first number is equal to a 1, for example, you have a 10% probability of winning the gamble, so generally you do not gamble. If the first number is a 9, then you have a 90% probability of winning the game, so you would definitely want to gamble if the number was a 9. So that's kind of how the game works. People figure out how it works pretty quickly. So the gamble option, again, is if the second number is less than the first, you'll get the $30, but if you're wrong, you get nothing. Okay, so we actually are in a position to be able to look at neural activity from microelectrodes implanted into humans' brains. And these are generally patients who had intractable epilepsy. So their medication is no longer working and they don't know, they can't control their seizures. So they come into the hospital for the possibility that a surgeon could take out the tissue that's generating those seizures. In order to do that, the neurosurgeon has to be extremely sure of exactly where those seizures are coming from. So those patients come into the hospital and there's lots of electrodes put in. Probably in the order of 100 to 200 recording channels are put into that person's head. And they have to stay in the hospital because all these neural recordings are going on. They're basically plugged into the wall and they're doing 24/7 recording to try to catch these seizures. So they're a little bored. So we come in and we say, "Hi, how would you like to play this game?" So while they're sitting in their hospital room, we have them play the game, and they're continuously recording their neural activity, so we can record their neural activity while they're playing this game. And these are from electrodes implanted in large parts across the brain in all of these patients. And then we just look at the neural activity that's coming out of each one of these electrodes that's implanted. Okay. So what we want to know is what about this neural signal is conveying information about the choice the person is going to make. So in order to do that, we're recording this local field potential, we call it. So it's in microvolts, so it's a potential recording. But there is a frequency component to all of these recordings. So you could take any one of these recordings and break it down into how much activity is sort of oscillating at a low frequency versus how much activity, so this would be a low frequency, or how much activity is oscillating at a high frequency. So for each one of these recording electrodes, we break it down into its frequency components. And then we take the activity around the time that the person makes the choice for the activity in each one of those frequency bands, and we ask, how does the activity change as the person's making their decision? Yes, we're going to align the data to the time of choice. So we're going to identify features of the data that are modulated by the choice. And we're looking at which frequency band. So is it slow activity in these recordings, or is it the very, very fast activity in these recordings? And where in the brain is this happening? So the features that are modulated, if we just look here and we ask the question, how many electrodes actually show some change in activity in each of the patients? Well, it's really only about 35% of the electrodes are actually participating in encoding the person's choice. The rest of them are just, there's activity there, but it's not involved. And then we ask, well, which frequency bands carry the most amount of, change the most during the task? And it really turned out that the higher frequency activity was carrying most of the information, and it wasn't really very much in these lower frequencies. But then if we looked how the information was carried across different brain regions, it seemed to be equally carried across all the brain regions. So all the brain regions seemed to be carrying some information, but it was always being carried in the high frequency activity. So if you just look at each of these are different areas in the brain that we recorded from, and then across here is the frequency band, and even across each individual region, you can see that the majority of the information is being carried in the high frequency activity. So those are then the features, the input that we want to use to try to decode what's going on, because this is where the information is. So information about choice is encoded in high frequency activity, and the information about choice is encoded across all regions of the brain. So we take this high frequency activity, and we're going to do two, what I call dimension reduction techniques on it. So remember in the beginning I said I had these hundreds of neurons and I want to find these latent variables in this sort of lower dimension. And one of the things I talked about and I showed an example of was principal components analysis, and how principal components analysis pulls out how the activity covariates together. And that was a pretty good representation, remember, of the animal pushing its paw up and down. Well, here we're going to use principal components analysis and see whether those latent variables that come out of that principal component can tell me about what choice the person made. And then we're also going to do another dimension reduction technique that I'm not going to give you too much detail about right now. I'll bring it up again later. But it's called dynamical systems modeling. And it basically just asks, if this is my firing rate right now, can I build a model that will predict what the firing rate is going to be at the next time? And you build this model, it's lower dimensional, so there's just a few latent variables, and we can use those then to try to describe how the brain is encoding information. So then we're going to take this, these latent variables that come out of these models, And we're going to do two different kinds of classifiers. So the job of the classifier is just to look at trials where the person made a safe bet and trials where the person made a gamble bet, and then take a trial, like a left out trial or an additional trial, and say, is it closer to the one where the person made the gamble bet, or is it closer to the one where the person made the safe bet? If my classifier correctly predicts which one it was, then the classifier is doing pretty well. If it makes a mistake, then the classifier is not doing that well. And so the performance of the classifier is how many correct trials was the classifier able to make. So it's going to predict the person made a gamble. If the person made a gamble, that trial is correctly classified. If the person actually made a safe bet, then that would be incorrectly classified. And we're just looking at the percentage of correctly classified trials. What we're going to do, two different kinds of classifiers. One is just a simple Euclidean distance classifier, where we just say, if this is the average activity, and this is the left out activity, I'm going to ask how far away are these from each other. So there would be one of these for a gamble trial and one of these for a safe. And then you would take your single trial and ask which one is it the closest to, just in a Euclidean distance sense. And then another is where we actually warp the time. And by this we mean we have the shape of what we think it's going to look like, but it doesn't have to line up exactly, and we can allow it to figure out, well, this is where this peak is, and this is where this peak is, and this is really the similarity. So it'll kind of stretch them and warp them in time, and it can do a better job, or potentially could do a better job classifying which choice the person made. And the reason we think this is important is because in some trials, the person makes a decision quickly, and in other trials, they take a little bit of time. So the question was, is the neural activity the same, regardless of whether you make your decision fast or slow? You just actually think it faster. So you run through all of the same neural computations and make the same neural activity, but when you make your decision quickly, you run through that neural activity quickly. When you do it more slowly, you run through it more slowly, and this dynamic time warping would allow us to line up those trials, even if the one was very long and one was very short. So we wanted to see if dynamic time warping did better, and then we would say, that might be because this is how the brain is working. So what did we find? Well, this is the accuracy. So this is the percentage of trials that the classifier classified correctly. And we have principal components analysis and linear dynamical systems, which I didn't tell you much about, but that's okay for right now. And the linear dynamical systems classifier always did better than when the dimension reduction was principal components analysis. And in fact, DTW under linear dynamical systems did better than Euclidean distance. So LDS, linear dynamical systems, outperformed the principal components analysis. Principal components analysis is saying is the information carried in how the neural activity co-varies together. And LDS is saying, I have a latent variable that can predict the activity at the next time step. So the variable that does better is the one that can predict the activity at the next time step. And then this DTW, this dynamic time warping, does better than the Euclidean distance classifier, suggesting that there is a need to kind of warp those trials to bring them all into line. So the pattern of the response is the same regardless of the duration of the trial. This is what the DTW is telling us. The other thing that was very important was that depending upon the win probability, or really, if you think about it, the first card, right? So if it's a 0, there's really no risk involved, because you should always take a safe bet, because no matter what, the next number has to be bigger, because there's nothing smaller than 0. And again, if you have if you get a 1, a 10, then you're definitely going to want to take the gamble bet, because whatever the next card is, it's definitely going to be smaller. So in some sense, when the first number is 0 or 10, there is no risk involved. And interestingly, you could not classify those trials. So the accuracy was 50%, which is chance, because half the time it's going to be a safe and half the time it's going to be a gamble. So we only got it right 50% of the time, which means we did no better than a random performance. Only when there was actually some risk involved were we able to decode. So we know that the information that we are decoding is related to risk. So trials with no risk, when probability is 0 to 100, can't be decoded. So we're not just encoding the decision itself, we're decoding the risk about the decision. Okay. So we are decoding risk. Okay. Let's see. Okay, so let me just go back for 1 second. Make sure yes, okay. So we talked a little bit about getting these latent variables, but I never told you what they look like. So this is an example of 3 latent variables from the linear dynamical systems model, showing how the activity changes with time, depending upon whether it came from a gamble or a safe bet. So one thing that's pretty easily noticeable is there's sort of this little oscillation going on here. And when there's an oscillation for gamble bets, there's a sort of an equal but opposite activity in latent variables related to safe bets. And that's true for kind of all 3 of these dimensions. And so if you think about them just kind of going back and forth between these 2, at the end, this is when the person makes their choice. They tend to be relatively separate from each other. So if we took these 3 latent variables that are here plotted as a function of time, but then put them into that state space that I showed you earlier, that is latent variable 1 as a function of latent variable 2 as a function of latent variable 3, and show you how these evolve in this sort of 3-dimensional space as a function of time, this is what it looks like. So I think I have to go here. So now I'm just plotting these 3 latent variables, but in this 3D space. Does that make sense? Okay. So they kind of come together and they cross, but they go back to their separate corners, and then they come more together and they kind of cross, but then they go back to their separate corners. And when it gets right around here, 500 milliseconds before the end, they sort of tend to start going away from each other and staying farther away from each other and sort of ending up in 2 different parts of that. If you consider all of this the state space, this is the neural activity kind of moving in that space, and they end up in their own little separate sections of that state space. So what does that look like? They really start to separate about 500 milliseconds before the person makes their choice. So in between 1 second before they make a choice and and a half a second before they make a choice, the latent variables are moving back and forth across the whole space. But then as you get closer, between a half a second to the time at which they make the choice, they sort of separate out into their own spaces and they don't go back together. So one thing you could kind of think about is that each choice is defined by a separate domain or space of this latent space by the dynamical systems model. So what you would have is that this would be the safe domain, and then this would be the gamble domain. So however the activity is being organized in the brain, the neural activity separates it into activity related to when they're going to make a safe choice compared to activity when they're going to make a gamble choice. So we wanted to quantify that separation because we want to know what is the rate at which it separates. So this example here is asking the question, if I have 2 of these trajectories in state space, what is the rate at which they run away from each other, or what is the rate at which they come back together? So if it's negative, they're going away from each other. If it's positive, then they're going towards each other. So initially in this 1 second to a half a second before they make their choice, They're kind of running more towards each other than they are away from each other. But then, as we noted before, about a half a second before they're ready to make their choice, they start to run away from each other. So this value, this Lyapunov exponent, becomes negative, and they are more likely to be separate from each other. This can be kind of imagined, the same concept can be imagined a different way, which asks how much do these domains here overlap as you move closer to the time of choice. And at first, again, in that 1 second to half a second before a person makes their choice, it moves kind of slowly, but then the overlap dramatically changes and they kind of quickly move away from each other in that last 500 milliseconds. So the person is sort of accumulating information about, do I want to make a safe bet? Do I want to make a gamble bet? Do I want to make a safe bet? Do I want to make a gamble bet? I'm pretty sure I want to make a safe bet. I'm going to stay in the safe bet side. Or do I want to make gamble safe, gamble safe? I'm pretty sure I want to gamble. I'm going to definitely gamble. I'm going to gamble. Okay? So from all of this, what we've been able to discover is that the information is distributed in this high frequency activity. So the lower frequencies are not really carrying information about what choice you're going to make. Decoding is improved by a model that's designed from a dynamical systems model, where the point of the model is to take the activity at the current time step and predict the activity at the next time step. And it's also improved by allowing the latent variables to warp in time, suggesting that when people make their decisions really quickly, the same activity is happening as opposed to on trials when the person responds more quickly, or more slowly, sorry. So each choice is encoded by a separate domain in this now latent variable space. So they separate from each other, and each latent variable visits each subregion during deliberation, but as time goes on, there's increased separation, which reduces the person making mistakes. So this last little piece that I want to shift to is those examples that I showed you, kind of here with this idea of can we begin to understand how the brain is encoding this information. That's the fun sort of theoretical part, but there's also a practical side to it in terms of can we actually develop systems that could help people who have pathological risk factors. So I showed you this psychometric curve before, basically that people tend not to gamble if they're confident the next number is going to be larger. They tend to gamble if they're pretty confident the next number is going to be higher. And different people have pretty much very similar psychometric curves. As these curves move to the right, you would be having increased risk aversion. And by that I mean, regardless of how much you were likely to win, you would still not take a gamble bet. So you would be very, very likely to take a safe bet for for lots of values of cards that you shouldn't have, right? You should have made the gamble bet, but you're going to go take a safe bet anyway. So your curve would shift to the right here if you were very risk averse. So let me show you an example of that. And the reason we care, I mentioned this before, I'm sorry, is that increased risk aversion is associated with depression. So people who are depressed tend to avoid making any kinds of decisions. What we then studied was, can I stimulate into the brain and change people's risk profile? So this is an example from one patient who originally in blue here, they had an extreme risk-averse profile. So for very, very many levels of win probability, they were completely unlikely to take a gamble bet unless it was like the card was an 8 or above, right? So they're very risk-averse, so their curve is shifted way to the right. Then we stimulated into the brain, and we were able to shift their risk curve to be less risk-averse. So the yellow curve here is their risk profile during the stimulation, and the green curve here is their risk profile 24 hours after the stimulation. So it doesn't seem as though you have to stimulate all the time. You could just stimulate, shift their curve back, and they would tend to stay back in that more risk-averse state. So this experiment just showed that it was possible to change people's risky behavior. So stimulation can move the state of the brain from risk-averse to a risk-neutral or closer to healthy behavior during and after stimulation. So we're engineers, and we want to do better than just some random stimulus. We want to actually identify the best, the minimal, the guaranteed kind of stimulation that you could provide at any time to shift the person's risk profile when you need it to. So if you think about driving a helicopter, okay, we design helicopters so they're man-made, so we know everything about them. But we design them so that they work in a linear regime. And all that means is that if I push the stick a little to the left, I'll turn a little to the left. If I stick it more to the left, I'm going to turn even more to the left, right? And I can pretty much interpolate in between there, and it's going to move in a linear way, which I like because my brain is simple, and it can understand that kind of linear representation, right? If I move the stick a little bit one time and it moves a lot, and another time I moved it, it moves a little bit, and it moved in some kind of non-linear fashion, it would be very hard for me to control, right? So I really want to live in that linear regime because I can predict things. And so it actually turns out to be kind of simple to do. There's a very much simple linear equation. So x at the next time step is equal to some constant times x at the current time step. So if these are latent variables that describe how the system works, y is the actual position of the helicopter at any given time, and y is just a function of these latent variables x. So this is exactly what we were talking about before with the neural activity. Can I get a latent variable that can predict the neural activity at the next time step from the activity of the previous time step? This is the linear dynamical systems model that I was talking about. So if in this case I have q is the x's here, these x's are either q, the pitch rate, theta, the pitch angle, or u, the horizon, and then you have this A matrix times the value of q, theta, or u at that current time step, and then this is the B matrix, and delta is the input I'm going to put to the system to make the thing go left or right or whatever it is that I want it to do. So this set of equations, which we've been using for, I don't know, a century almost, to build systems that we can control, we're going to try to use this to try to control the brain system. So I could stimulate into the brain, which would be the U in this case. I can tell you what the current neural activity is, and I can use it to try to predict the latent variable at the next time step and tell me how to stim in order to make your activity at the next time move farther away from or more towards being risky. So since I have a linear model, I can guarantee what's going to happen, and I can be very certain of how much I can stim. But the question for us is, can I actually build this model from neural data in the brain? So the idea is that I have this regime in the brain for safety, making safe choices. I have this regime in the brain where I'm more likely to gamble, I'm more likely to take risk. And if I'm sitting too long in the safe space, I want to stimulate and move you to the gamble space. If I'm sitting too long in the gamble space, making bad decisions, I want to shift you back and put you more towards the safe. But I want to keep you in some sort of neutral territory. So we can record the person's choice, we can decode the output, we can put it back into a controller that says whether or not we should stimulate to change the way the person, where the person's space is. So we're going to build a control system that continuously decodes the state of the brain regarding the tendency to gamble, and will transmit appropriate information through microstimulation to reduce the likelihood of gambling. I don't know what happens to that word, but, or in our case, we're doing it to increase the likelihood of gambling. So for you guys who like engineering, this is kind of the representation we would have. And basically, we have the brain, we record the neural signal, we decode the current state, where does the person live in this state space. We have this calculated state of where the brain is. We have a desired state. We don't want them to be too over in the safe side. We don't want them to be too in the risky side. We have a desired state. And then we have a feedback controller that says, what's the difference between the current calculated state and my desired state? And then that tells me how I should stimulate back into the brain to change how you are. So the advantage of the linear model, as I said, is that it's the outcomes are guaranteed. They have to work in this linear system. So this is just kind of a little cartoon to kind of re-emphasize what we're going to do here is that we have the brain, we have a function u of t with how we're going to stimulate the brain. We're constantly recording the person's intracranial activity. We're going to build a model that can predict the neural activity. If it's a good model, then we will have be able to at each moment in time, you'll know how you stimulated and what the current state is, and then you'll be able to decide how you should change the stimulation to change the state. But the first thing we have to do is check that we have a good model. The way we check that we have a good model is that we can predict what the stimulation, what the neural activity is going to be in response to stimulation. So this is work done by Ariana Morvell in my lab, and basically this looks a little more complicated than it really is. We are continuously recording this high gamma activity, so this high frequency activity, and we're going to stimulate, and we're going to record the response to the stimulation, try to build a model that predicts that activity, and then test how good a job we did on that. So in this dark sort of teal line, it's the true response. The black line here is the model response. And we're doing 3 different kinds of stimuli. We're doing stimulus at one channel, stimulus at a second channel, and a stimulus at a third channel. And so it's very hard to see this. I totally appreciate it. So we just sort of blew it up down here. And so again, the teal is the true neural activity. And the thin black line here is the model prediction. So we take this activity, we build the model, and then we test it. And what you'll notice, most importantly, is that our prediction almost always overshoots what the actual system is doing. And this is very typical of linear models, because anything that's changing very fast is going to take probably more than a linear model to reproduce. So it kind of overshoots and then it comes back. But it's not a bad job reproducing the activity, right? There's definitely a little undershoot and a little overshoot, but in general, it's getting a fairly good job of reproducing the neural activity. So that's the step 1. Can we build a model, and can that model reproduce the neural activity? Then we want to ask, how would we stimulate in order to push it from one direction to another direction? So, so here, the effective stimulation can't be predicted, and that basically tells us we have a pretty good model of the system. So here's an example where we stimulated in the same location in the brain, but the brain state was in a different initial condition. So here are a bunch of stimulations. Each one of these arrows is a stimulation, and in one case, these were all in this place in the state space, and every time we stimulated, it pretty much went in the same direction. And here's another set of stimuli. The tail of the arrow is where it starts before stimulation, and the head is where it was at the end of the stimulation. So here's an example of a bunch that were in one state here, and they pretty much tend to go into this direction. So it's almost as though you could imagine a boundary here where everybody on this side is going towards this sort of little region in the state space or attractor, and on this side, everybody's more likely to push you towards this one. So this gives us some idea that we could probably control the outcome of the stimulus if we know the initial conditions and we pick the proper location to stimulate. You could so the effect of stimulation depends on the initial conditions. So here I'm doing the same stimulus, but depending upon where the activity of the brain was at the start of the stimulation, I'm going to get a different output. You could kind of think of it the other way around, where now I have the same initial conditions, but I'm stimulating in a different location. So here, all of my starting points at this end of the arrow were all kind of in the same location, but for the purple, I did a stimulation at channel 1. For the red, I did a stimulation at channel 2, and for the green, I did a stimulation in channel 3. So even though I'm starting in the same location, I can move the neural state to a different place. Here's another example where everyone is kind of starting in the same place, but if I stimulate at channel 1, I kind of go in this direction, and I stimulate a pulse at channel 3, I sort of go in that direction. So similarly as being able to stimulate in the same location, but different initial conditions, the stimulation also depends on the stimulus location. So we now know that it's possible to continuously record the neural activity and predict the person's risk state. It's possible to stimulate back into the system and modify their brain state. So it's possible to predict the outcome of a single trial of choice. During deliberation, low-dimensional latent variables move through these separate subspaces depending upon the options. And response to stimulation can be predicted, and it depends on both the initial conditions and on the stimulus location. So this is actually kind of where we are at this point, and what we need to be able to do now is go back in while they're doing the task and stimulate and see if we can change their choice on each individual trial. So we're gearing up to start to do these experiments while we'll decode what decision the person's about to make, and then try to stimulate and see if we can change what their outcome is going to be by using this feedback control system. So it tells us that building a control system is likely possible. So I just want to thank my collaborators. So I mentioned earlier Ignacio Sayas, who's a professor at Mount Sinai, who's doing the task. Dr. Jiadong Kong is a colleague in the aerospace engineering department, and he helps a lot with the development of the control system. And Dr. Jing Liu is in computer science, and she's been helping with the classifiers. So couldn't have done any of this work without them. And then the 3 graduate students that I mentioned during my talk. Alec Rodemel did a lot of video analysis of coding. Logan Peters is looking at a lot of nonlinear analysis of coding. And Ari is working on the behavioral effects of stimulation. So thank you very much. I hope I kind of kept you guys with me, but even if not, you can feel free to ask a question about anything related to this. So it doesn't have to be necessarily what I said, but if you have any interest in brain-machine interfaces or how these neurotechnologies are going or what's going on in the world with that kind of stuff, I'd be happy to answer any questions about that too. So.
So we're going to give people 5 minutes. So if you want to stick around for questions, we'll start that in about 5 minutes. And if you're just ready to go home, then this is the time to do that or sign in. Questions in 5 minutes.